The Mesothelioma Genome Project aims to better understand the molecular causes and characteristics of malignant mesothelioma through comprehensive genomic analysis. However, this ambitious research initiative faces several important challenges. One major challenge is acquiring high-quality genomic data from a sufficient number of patient samples. Malignant mesothelioma is a relatively rare cancer, with only around 3,000 new cases diagnosed in the United States each year. This rarity makes it difficult to obtain large datasets needed for robust genomic profiling and association studies. Researchers must collaborate across multiple institutions worldwide to assemble sizable sample cohorts. Issues like sample degradation over time and lack of detailed clinical data can also reduce data utility. Surpassing a critical sample size threshold is crucial for reliably characterizing the genomic landscape of this disease. Another challenge lies in the molecular heterogeneity of mesothelioma tumors. Evidence indicates mesothelioma exhibits substantial intratumoral genomic diversity both between patients and within individual tumors. This diversity complicates efforts to identify consistent mutational signatures, recurrently altered genes and pathways, and reliable biomarkers. Integrative analyses involving multiple omics platforms, genome, transcriptome, methylome, etc., may help discern the underlying biological complexity but require tremendous computational and statistical resources. Distinguishing true driver alterations from passenger changes against such a backdrop of heterogeneity also poses challenges. Analyzing genomic data from mesothelioma poses certain technical difficulties as well. The tissues utilized for DNA and RNA extraction often have a large non-tumor component containing normal pleura, fat, or germline cells. Contamination with non-neoplastic material can dilute cancer-specific signals and hamper discovery of subtype-defining alterations. Methodological choices around sample procurement, histopathological evaluation, and purification of neoplastic cells influence data quality and findings. Achieving sufficient purity from small biopsy specimens can be problematic. Clinical and phenotypic annotation of mesothelioma samples also presents challenges. Many tumors lack robust information on variables like asbestos exposure history, treatment response, and survival outcomes. Integrating genomic findings with clinical parameters requires thorough retrospective or prospective data collection from multiple centers. Factors like inconsistent staging practices or absence of standardized exposure documentation hamper meaningful integration. Developing prognostic or predictive models also demands well-curated clinical cohort resources which are difficult to assemble for a rare disease like mesothelioma. Bioinformatic analysis of massive genomic and clinical datasets poses substantial computational challenges as well. Techniques for detection of subclonal variants, transcript reconstruction, integrating multimodal omics profiles, and statistical modeling with many variables demand considerable processing power and algorithmic sophistication. Storage, management and sharing of resultant petabytes of data need robust infrastructure support. Developing user-friendly interfaces permitting interrogation and reuse of generated resources requires ongoing software development efforts. Meeting evolving community standards around data access and participant privacy also necessitates active engagement. Validating research findings and translating results into clinical utility faces roadblocks. Confirming candidate genes, pathways or biomarkers identified through genomic studies necessitates independent experimental validation, often requiring fresh samples. Developing and testing therapeutic strategies based on insights into disease biology and subtyping also demands long lead times and substantial coordinated work across fields. Regulatory approval pathways for new diagnostic tests or targeted drugs are arduous. Demonstrating an impact on patient outcomes constitutes the ultimate challenge. Overcoming these barriers to establish the clinical relevance of genomic knowledge will prove very challenging. Due to the rarity and complexity of malignant mesothelioma as well as limitations in biopsy resources, clinical data and computational capabilities, the Mesothelioma Genome Project faces significant scientific and logistical difficulties. Surmounting these challenges demands enormous collaborative efforts, methodological innovation, new computational frameworks, and sustained commitment of expertise and infrastructure support over many years. Success would undoubtedly advance understanding and clinical management of this lethal asbestos-induced